Good afternoon, everyone. We're going to get started. My name is Mustafa El-Shahed, and I'm an associate professor at Oklahoma State University. And this session is about the microbiome, okay? This is session A5, the microbiome, a new way of understanding our world. And we really have three excellent speakers. This is the caliber of speakers and the type of speakers that you get in the best international conference ever there. So if you're a researcher here in Egypt, please take this opportunity to listen to all of this new and exciting science that we have. And we are going to start with Dr. Rami Aziz. My dear friend, Dr. Rami Aziz, graduated from the same university as me, from the Faculty of Pharmacy, Cairo University. And he is currently an assistant professor, if you're Egyptian, or associate professor, if you're somewhere else, at the Department of Microbiology and Immunology at the Faculty of Pharmacy, Cairo University. And Dr. Rami Aziz has many research interests, and I can't say all of them, but he's an expert in bacterial pathogenesis, in microbial genomics, in phage genomics, mobile genetic elements, transposons, all different kinds of metagenomic and bioinformatic binding. And he's also a very active and avid user of social media. And he is interested as well in scientific communication, open access publishing, and finding new and innovative ways to make science interesting for students. So without further ado, Dr. Rami Karam.
Thank you very much, Dr. Mustafa, for accurately describing me as a jack of all trades, master of none. In Arabic, سبع صنايع. So we did a little switch in the program. I'm going to speak before Dr. Mira because I'm going to introduce, half my talk will be just introductory of what is the microbiome and the human microbiome. And my title is Our Microbiome and Us, A Common Destiny. I would like to start with, since Mustafa already referred to, I have changed interests over time, and because this is relevant to the talk, I'm going to just let you know. I started mostly working on medical microbiology, microbial pathogenesis, interest in group A Streptococcus, and this led me to study the phages in group A Streptococcus, which moved me, without me feeling it, to microbial ecology, about which I knew nothing before, through genomics and bioinformatics. So the bioinformatics of the phage led me there. And I did some little work in this when I was doing some postdoc in San Diego. And then it was just the time the microbiome project started, and the reason I'm telling you this introduction is microbiome is the reconciliation between ecology and medical microbiology. There was a fake division in microbiology that people say, oh, I'm studying medical and the other is studying ecology, but it's really the same field. The only difference is that human is the ecosystem, and we are not different from any garden or mountain. We're just a good place that microbes eat. So the outline of my talk will be, first, a little bit of introduction, and if I were not a scientist, I would have liked to be maybe interested in philosophy. So I would talk a little bit about how to describe the microbiome, because this matters a lot in how to study it. And then I'm going to talk about two parts of research I have done, partly bioinformatics and how to decode the microbiome, make sense out of it, and what are the problems and bottlenecks, which Bent will talk much better than me about later. And then a little bit about the effect of microbiome on human health, disease, and therapy, since I'm in a pharmacy school. And before starting, and before I get too busy at the end and get forced to finish the talk, I would like to thank people who did the actual work. And many of them are students of pharmacy school, Mariam, Rama, and Mariam. Abdulrahman is also in Bani Suwaif, and I'm just co-advising him, but he did great things. Sherry and Ali from the American University, with collaboration of many people around the globe. So to start with, what is the human microbiome and what is HMP, or the Human Microbiome Project? The whole story started in 2001, kind of, when it was announced that the human genome has been finished. by two groups, the Venter group and the entire world. So it was Venter ahead of the world. And the big surprise is, which we heard then in 2001, we're so excited, the human genome has been sequenced, we have the entire code, so we have the solution to all problems. But you know from all the talks yesterday and today that we're far from knowing the solution. First of all, the human genome is really hard to decode. Dr. Ahmed Mustafa has now been mining 10,000 genomes, and we're still trying to understand. But what we're really seeing is that phenotypic differences are not explained by the human variations. We are very similar, after all. Even with all these SNPs, people are very, very similar. So there must be a lot of regulation, as you heard in the morning, epigenetics. But then the missing part was the microbiota. And luckily for microbiologists, they got us involved then. It was called the Human Genome Project, but slightly it was converted to the Human Microbiome Project. So this is good news for microbiologists because it means work to do. So first of all, what is this term, the human microbiome? Scientists like to make new terms for old things. So we always heard about the normal flora, which is a very bad name for bacteria. It's not fair because bacteria are not plants. So the normal flora, or the right name is the microbiota, is just the bacteria that live in and on us. We are actually just, we are very important. We're human, but to the bacteria, we're just food material. And we are a big tunnel for them. So we have a gut that starts with the mouth and ends with the anus. So they like to live inside, and also they like to live in anything else they can live. So the skin, the vagina, the hair, nares, and so on. And this made the news a lot in the past 5 years. This was The Economist, not a scientific journal, and they talked about how men are made of microbes. A little bit of media exaggeration. And this number you see a lot is that the number of our cells is about 10 to the power of 13. Some people argue we have more cells, counting the red blood cells as well, which don't have a nucleus, but more or less we have that big number, 10 millions of cells, and we have almost the same number of bacteria, a little bit more. There is a lot of arguments we don't care about, is that who is more. So bacteria, for every human cell, there is 2 to 5 bacterial cells. So they outnumber us. And then the only part I told you that I will get into a little bit of philosophy, or images are very important. So when you see this image, you feel like we're made of microbes, but it also gives a little bit of false impression. So I would like to, there is no time for me to take a vote, but I will show you a few pictures that came out in the news to represent what the human microbiota looks like. This one, and then this is the most classic one, like we have bacteria in our nose and so on, but it's very human-centered. And so on. You see a lot of pictures like this. I might ask you to vote which one is your favorite. You can vote secretly. But here is another impressive one. And so on. These pictures make a lot of difference when you study the microbiome. So the most common picture, or the most common way to describe the microbiome, which Alex and I might a little bit argue about, is the most common way is to say the microbiome is a forgotten organ. So we have a kidney, a stomach, and we have also a microbiome, an extra organ. And I don't like this very much because it's not a single organ, it's a lot of bacteria, they're everywhere. But more importantly, it's a cloud of uncertainty. Like our stomach, we know the cells, they have some, doctors know better than me about these, physicians. But the bacteria change all the time. So it's more like, you know this, right? It's the electron cloud model. It means that we're not certain at any point where the electrons are. I would argue that microbes are more or less the same. We have a cloud of bacteria. They change all the time. They divide. If you just go to the toilet and come back, you have lost a big portion of your microbiome. And then you eat again and you reinstitute it. So there is a lot of exchange. And this is, we knew this all the time. But now it's exciting because they affect our health. You give somebody a medicine and you want to treat them. So you want to know that there is the missing organ is actually a missing cloud. There is some extra component here, which changes over space and time. And when you're young, it's different. If you're born cesarean, if you're born naturally, you have a different microbiome and so on. Depends in women on the hormonal stage. So we're surrounded by these microbes. Not only we don't know all of them, even if we have the means, the moment you know them, they change. So there is a lot of change. This is the end of theory. So just my first message is that we are really inhabited by a cloud of microbes, which means a lot of genes and genomes. And they have genes, they express proteins, they have action, they have enzymes, microbes and their viruses, and we're kind of domesticating them. We're living friendly for centuries and for thousands of years. But this cloud enriches our gene pool. We cannot live without them, actually. We cannot digest a lot of food without them. We need vitamins. They protect us from competing microbes that are called pathogens, and so on. So this is why the microbiome is really exciting. You will never finish studying it. There is always new. All right, so once I define the microbiome and the way I see it, I want to tell you a little bit, very, very quickly, a few examples of problems in understanding actually or decoding, reading the microbes and what genes they have, and how we, like in collaboration with many other people, we try to solve this. The first problem is actually before even the microbiome. It's the problem that you heard about a lot today and yesterday. It's decoding the genes. So it's like going to an old manuscript, collecting pieces and trying to decode. It's hard to assemble them, and probably Bent will say something about that, but it's also, once you have a genome, a lot of A, G, C, and T, it's really hard to know what they do and what they do together. So the first effort I was luckily involved with a very big group in the US and UK as well, is developing a way to annotate, which means give name to the genes, try to know what they do. And we created this so-called the RAST server that tries to use subsystems technology. It's just a way of annotating genes that was much faster. Before that, to get one genome annotated, it took months and months. And after RAST, at least you get a draft automated computational answer in 48 hours. Now it takes 2-3 hours. And this is the way it works now. This is taken 3 days ago. So this is how many microbes are sequenced every day and annotated. So just look at this. This is last Monday, midnight to noon. All these jobs are uploaded to RAST. And, oh, sorry, this says 2014. It's a mistake. It's 16. And all these were uploaded as well. The other problem is the so-called mobile elements. So there is a lot of genes that move inside the genome, and many of them are called transposases, but there is also plasmids and insertion sequences. They're very much troubling because you have, it's like you're reading a book, but there is a page that changes every copy of the book, you know. It's like a surprise page, or like find ابحث عن فضولي or something like that. You find a different, so these genes, when they move, they might inactivate genes or add functions. So we worked on the transposons a little bit. It's really hard to find them because they're everywhere. So we tried to clean up the annotations, try to find the sequence, and we found out that they are the most abundant genes in microbial genomes. But what they do, they are so-called selfish genes, which means all their purpose is to replicate themselves. But again, philosophically, we're all selfish genes, right? We all here to survive. But in fact, they also generate a lot of functions. Just an example, our immune system is edited. It creates variability by a previous transposon gene that was transformed into a recombinase. So these genes are everywhere, even in nature, and they have a lot of function. Then the other component is phages, and I'm really glad I'm talking after Martha, Dr. Martha Clokey, who introduced what a virus is, a microbial virus. Phages are very, very abundant, and we found them a lot in the human microbiome and in any other microbiome, in the Red Sea microbiome, in the soil microbiome, and it's really hard to annotate them as well. Again, I'm just trying not to distract you, but we're trying to understand what these microbes do and what are the mobile elements do and what their functions are. So it's really hard to quantify viruses. You heard that bacteria, we can just tell what bacteria species is or genus from a 16S gene. It's a small gene that produces a ribosome, and we can tell from that sequence what the bacterium is. In viruses, there is no such thing. So you have to look at the entire genome. There is no single gene in bacterial viruses that is common to all of them, so you cannot compare. So I worked again with Rob Edwards and Maya Breitbart in the US on developing metrics to get what phages and how many of them in a certain microbiome. Now there is the assembly problem, and Bent will talk a lot about it. But again, I borrowed this picture a long time ago from Dr. Kropinski. You have a genome, but if you do a mistake, for you, it's just, you swap the page of the book, but it creates a different creature. Like this is a virus. If you read the genome or you assemble it wrong, you will get something totally different, right? It's not the same. Microbiome is even different because you're sequencing a lot of microbes in the same time. So it's like you went to a forest and you found this ear of a rabbit and this corn and these feathers and so on, but you were too ambitious, you used the computer and you don't know anything about animals and you get this. So it's very common. It sounds like a joke, but everyone sitting here knows that you see these. You make a new virus that doesn't exist because you assemble two parts of a wrong virus. And the reason is always these repeat sequences I talked about. The best example is The Tale of Two Cities. Have you read The Tale of Two Cities? It's a book that has, in the first page of it, it was the time of wisdom, it was the time of power, it was a lot of repeated words. So if you cut the book into pieces and try to read it again, you will find the same word repeated many times that you won't be able to arrange it. So I have a very smart collaborator in Netherlands. His name is Bas Dutel, and he developed a way he called it cross-assembly, that's the way, crass, and we discovered a completely new virus that is in every other person. So if you're seated here, one of two of you has this virus in their intestine. We called it crassphage because it was assembled by cross-assembling. We could never find this virus because we didn't know anything about it. So every time we find some sequences, we say we don't know anything about them. What Bas did is he assembled all the genomes together, all the samples together. So he took samples from like 100 persons already published and tried to assemble something from them, a little pieces from here and there, until he found a full virus. We couldn't find all the pieces in one person because it's fragments and undersampled. But once you find it, you go back and you find parts of it in every person. So 50% of humans have this virus, and it was published a couple of years ago. We still don't know much what it infects and what it does, but many people are working on it. The best thing about it in applied science is you can, since it lives in the intestine, if you find it in water, you know that it's fecal contamination, for example, or sewage contamination, so at least it has a use. All right, one more bottleneck and a problem of analysis that Dr. Krupinski talked about on the first day. It's finding antibiotic resistance genes, which is really important for many of us. So people would go to the, again, microbiome, sequence a lot of DNA from intestine, from the ear, from anywhere, and you say, oh, there is 10 resistance genes here, which means there are bacteria that carry resistance genes. But you also find them in unexpected places, like caves under the water and things like that. What are they doing? We are very, anthropocentric. We're only thinking about ourselves. But before antibiotics were discovered in 1925, bacteria were still producing these, they didn't call them antibiotics, but these weapons to kill one another. So any bacterium would have a weapon and other bacteria will have a way to deactivate it. So you find them a lot. The problem is that many of these genes were misannotated again. They were not, for example, if you find a DNA polymerase or RNA polymerase, this exists in every bacterium. You cannot say this is a resistance gene. But if you go to the CARD database that many of you use, it will tell you this is a resistance gene because if it's mutated, sometimes the bacteria would resist rifampicin, for example. But this is not real resistance. So we just fixed the way it's analyzed. A very smart student, Ali al-Bahiri at the American University. It was just published last month in collaboration with Rania Siam. We did this pipeline and it was published 2 weeks ago. Rania is an application of all what I said, trying to look at microbiome that is actually not human in this case. Rania is very interested in the Red Sea microbiome and she collects a lot of samples. So we took some of these samples and one of her students applied the method I told you about to find viruses, bacterial viruses, and this is work in progress, but she's now found that 2 of the 10 sites in the Red Sea have very different, all you need to see is that this is different from the rest, right? Can you agree with me? This is dark and light here, and here it's everything. This means they have underrepresentation of a lot of viruses, and these are the deepest layers. At the same time, Ali discovered the same thing. If you go deep to these 2 layers, they are also enriched in mobile elements that are not viruses, like insertion sequence and transposons. So we're trying to understand what this means. And what you need to know is that the bottom of the Red Sea is full of heat. They call them hydrothermal vents. It's like a volcano. an extinct volcano, but it still has heat. And many people argue that the origin of life on Earth was similar to this. So once you find a lot of insertion elements and transposons that actually cause variation, you're tempted to think maybe this is a good thing when you have an environment that is very old. So you need a lot of variability. And once you have diversified a lot of bacteria and viruses and things like that, you start slowing down. You don't want that much variation. This is a very ambitious argument. There is another argument, is that just the type of bacteria that live here, like halophiles and other bacteria that we call extremophiles, they are just known to be enriched in these elements. But again, why? We think it has to do with their lifestyle. They might be very beneficial for these bacteria. Anyway, so I conclude my second section here. So first I try to define what the microbiome is, and then now I talked about how to decode it. And many people in the microbiome field think that the most important thing is to know what bacteria are there. For example, look at your intestine, you have 10 types of bacteria. But what really, I would argue also, people always tell you in sociology that who you are is more important than what you do, and this is true in sociology, but in bacterial biology, in microbiology, it's not that true. Actually, it doesn't matter who you are as a microbe as long as you have the same enzyme. So this argument, there is a big debate here. And actually, I found out it was there 100 years ago, in 1906. Somebody published a paper that, 1909, sorry. They said the exact same thing, that the fecal mass in our body is made of bacteria. And just to cut short, he said, it is what bacteria do rather than what they are that commands attention. So from our point of view, it doesn't matter if you have a streptococcus, a lactococcus, or a bacteroides, but the question is, do they break carbohydrates down, or do they actually, they're more proteolytic and so on? Because this is what will give you either good health or cancer. So that's what we really care about. So when we just look at 16S, we miss a lot of functionality. And I don't know, maybe the other speaker will agree or disagree, and then we will have a debate at the end. So my second message is that decoding the microbiome is an art and science, which means it still needs a lot of non-transferable skills. You still have to tweak some things. And this is a very good field for everybody. There is not an answer to every question in decoding genomes or microbiomes. It's still a good field to study, and many of you I think, are working on this. So we need to think outside the microbiome box, try to assemble outside and find some answers. Then it's really important to ask a good question. And I was discussing this with Dr. Mustafa a few times ago, last week and last year, is that sometimes you start a project, it's really easy to go to a forest and describe what the plants are, but then you don't know what you want to do. So it's really good to have a question in the beginning. For example, is this bacteria good for my health or not? It's not just, oh, I have 200 types of bacteria in my stomach. It's exciting, but then what? Both are exciting. I don't argue against this or that. Last but not least, I want to talk about how the microbiome affects our health and disease. One very well-established model, and it's already answered, is that there is a balance that the bacteria cause for example, in our gut or in our nose. And once this balance is broken, you don't have a disease yet, but you reach a state that they call dysbiosis, imbalance, that give the chance for external pathogens and internal opportunists to take over and grow more than they should grow. It's something like cancer, but not exactly. But these bacteria, when they grow out of proportion, they cause a disease. And the best example you heard is Clostridium that causes diarrhea sometimes. It also affects our nutrition. So some mice are fatter than others just because their microbes like to deposit fats in their, So in some sense, as we say in Arabic, هذا ما هجناه microbiomi علي. It's not really our fault. And then there is also a cancer model well developed, and I think I will skip that because I believe Dr. Alex Mira will talk more about that. So some bacteria are associated with colon cancer, and actually they have causality. Now I want, just before finishing, to give you two examples of my ongoing research. The first, I found the best thing is to put Mariam's picture with her poster. It's really clear. So Mariam, who is sitting here and she has a poster, is studying the difference between normal eyes and diseased eyes that have conjunctivitis. If you try to, so many people would go, somebody has conjunctivitis, they separate it into bacterial and viral. If it's bacterial, they take a swab of your eye and say, oh, you have Staphylococcus aureus, this is why you're infected. And we thought, well, maybe there is Staphylococcus aureus here too. If you go to one person who is infected and another who is not, there is really a lot of variability, and this has been found in all microbiome studies. So if you sequence 10 people, you'll find too much variability that you cannot really say anything. So we just thought, okay, everybody has two eyes. Let's collect only the patients who have conjunctivitis in one eye and still the other is intact, okay? And let's compare them to some normal people, and Mariam was so courageous that she also swabbed herself. And then the next slide is really crowded, but I will tell you the message, okay? So each one of these bars with all the colors represent one eye. So these two eyes from one person, non-infected, infected, non-infected, infected, and so on, non-infected, infected. And these four are controls. They don't have, both eyes are okay. What you see is, first of all, as I said, inter-individual variability is much higher. So if you take 3 persons that are normal, they don't have any infection, too many different types of microbes. So you see very different patterns of colors. But you also see this pattern. So here is Staphylococcus aureus. It expanded in the infected eye. Here is another infected eye that almost doesn't have Staphylococcus. It has less, and so on. So we try to follow this, and we're still working on the analysis. But the main message here is that probably it's a dysbiosis question, which means you have a lot of bacteria in your eyes. One of them might take over out of proportion, and it might cause conjunctivitis. So we call it an inside job. We're still not 100% sure of this hypothesis. We're trying to prove it or disprove it. But this has been seen in many other things, that sometimes the disease is coming from within. It doesn't have to be an external factor. So to recap, we have clouds of microbes around us, and what they do is not always correlated with who they are. The last thing I want to tell you about very quickly is, because we are in a pharmacy school, so we got interested in this. Do they also affect the drugs we take? And the answer is yes, they do. So bugs versus drugs. So you have, just think about it. You have, a drug target, which you're trying to treat this person, but the person is surrounded by a cloud of microbes. And these microbes produce enzymes, and some of these enzymes would break the drugs down. I will skip this, but just think about the sun with a different size of clouds. It makes different effects. So a drug will also make very different effects on us. So I'll skip this for the time. Basically, the bacteria in our gut may affect the drugs we take directly, degrading them, or they may produce some metabolites that compete with them, or they may even affect our liver enzymes, which anybody who studied pharmacy knows that these are the best protection for us from the drug toxicity. So if the microbes have all these effects, they can definitely affect the drugs we take. We're trying to study this in the lab, but before doing this, we went to literature, and we found that there is a lot of old examples from plant biology and from pharmacy, but nobody has a database. So Mariam and Rama created a database online we call the PharmacoMicrobiomics portal about 5 or 6 years ago now, 5 years ago. And we're having good traffic. So this database, you go and find about 70 drugs, medicines that are already known to have bacterial effects. So the last example I will give, it's an ongoing research as well. Very ambitious student, Abdurrahman Ali from Bani Suif. He just asked me to help with his supervision, so I'm not his main supervisor. And Abdurrahman had access to hepatitis C patients. He collected stool from 6 patients and 8 healthy individuals. This is a lot regarding our budget. And on his own, he contacted a lab in the US, very big lab, PNNL, and Dr. Janet Johnson very generously accepted to sequence for him for free. You know her name now, she will regret it. So what Abdurrahman found very quickly is that, again, this is healthy, taking treatment, the triple treatment, and then taking treatment for 3 months and so on. So you will find things that increase after the treatment and less in healthy and vice versa. I think I'm out of time, so I will just say that 2 bacteria differentiate the hepatitis from the normal, and they are gut bacteria. And we have a lot of ongoing projects. Many of the students sitting here do something related to microbiome. But yeah, for fairness to other speakers, I'll skip that, and I'll just give you my final message. Our cells are surrounded by a microbiome cloud. Think of the microbes around us, that they are very variable. We cannot know exactly who they are at every point, but we can get an idea, a core microbiome. They affect our nutrition, health, and disease, and therapy. And decoding them is a challenge, but it's a call for everybody to help. It needs a lot of work, manpower. Finally, our future is eternally connected to our microbes, the intimate strangers, and we have a common destiny. Thank you for your attention, and I'll take any questions. Burning questions.
Yeah, thank you very much, Rami. An excellent start for the session. Any questions for Dr. Rami? Yes, there's a question here. Oh, okay, yeah.
First, I'd like to thank you for your talk. At the first, when you talked about human microbiome, I was very interested, and I thought it would be a new era for the understanding of ourselves. Then you said that there are similarities between human microbiome and microbiome elsewhere, and then I got confused. What do we conclude from these similarities?
So that's a good question. Maybe I didn't express it really well. I said that the same way we analyze them, we analyze the Earth microbiome, the Red Sea microbiome, and our microbiome is very similar. So it's the same way to analyze. But actually, you're also right. So many people actually think that we are very similar to what we eat. So there is a big, the diet makes, especially the intestinal microbiome is the richest in us. So it's very much affected by what we eat. And there is a very good science done by Professor O'Keefe in the UK. You should read about it. He got people from Africa and African Americans living in the US, and he swapped their diets for 3 weeks, just 3 weeks. The people living in Africa, they would eat burger, and the people living in the US from African descent would just eat, later you will discover it's healthier food, rich in fibers and carbohydrates, no much proteins. And in 3 weeks, the tumor markers were inverted. So those poor guys who started eating burgers, they developed tumor markers and for colorectal cancers and vice versa. The good news, it's curable. I mean, it's reversible so far. So you can change your diet for a while and fix it. So there is a lot of similarity. And for example, the eye microbiome is very similar to skin and nose. So things are similar to what you touch. Just to save time. Another study was done that if you go to a new house, after 3 weeks or 4 weeks, you transfer bacteria to that house. So there is exchange between our microbiome and other. And some people would even go to say that if you leave a place for 5 minutes, you leave a little aura around you of microbes. So there is a lot of things going on. Just to briefly answer your question. Thank you.
Okay. Another question?
Yes, please.
Down here.
That's a very good question. With two people living in... so would two people living in the same village develop the same microbiome? It depends on which microbiome. So the eye microbiome actually is very much affected, even in Giza, so where Mariam did the study. It's affected from which village people come or what job they do. The skin and eye microbiome might depend on that. The gut depends more on food. But also, I don't want to spoil Alex's story, so he will have you're not talking about that? I think part of Alex's story, he will tell us that how people may exchange microbiomes, right? Yeah? Okay, so I mean, I don't really want to take more time because at some point they will tell us your time is up.
Rami is always here, so feel free to approach him. Okay, so moving on, our next speaker is Dr. Alex Mira. Dr. Alex Mira is from Spain, and currently he's the principal investigator in the Oral Microbiome Laboratory in the Advanced Research in Public Health Center. Dr. Mira got his bachelor in 1994 in Alicante University in Spain. Then he did his graduate studies, both his master's and PhD at Oxford University in the United Kingdom, where he was a Rhodes Scholar. And Rhodes Scholarship is an extremely competitive, extremely prestigious scholarship that only few people in the world really get. And he just told me he was the first person from Spain to get a Rhodes Scholarship. He then he obtained a Fulbright Scholarship, which is another very prestigious scholarship, and he did his postdoctoral work in the lab of two very prominent scientists, Dr. Nancy Moran and Howard Ockman in the United States. And then he went to Sweden with Dr. Siv Anderson, at which he did a lot of interesting research. And then in the year 2003, the government of Spain decided it's time to put some incentive to induce great scientists from Spain to come back home and help the country. and he did return home in 2003, and he's been doing research then ever since. And while he was in Spain, he obtained different types of awards from the government of Spain, from the Spanish Society of Microbiology. He published more than 80 publications in international journals, including Nature, Trends in Genetic, Nature Reviews in Microbiology, and many other great journals. And I will stop here and let Dr. Mira start. So.
Wow, thanks for the introduction. It sounds much more than it actually is. Yeah. I'm really excited to be here. I'm very, it's a big honor for me to be here at this special venue. I'm very grateful to the organizers and especially Dr. Rami for the invitation. And they also make me feel at home, even more in this city where everything appears to be called Alex something. And I have realized I even have my own bank, the Alex Bank, last night. So it's a very big pleasure to be here to tell you about the research we have been doing in my lab in the last 5 years. And I'm going to focus mainly on the clinical implications of the work that we have been doing. It's funny, most of the talks on the topic, they always include 3 sentences that everybody says. The first one, probably you have heard this before, is that we have between 1 and 2 kilograms of our body weight is bacteria. I mean, how did they measure that? I mean, does anybody in the audience know how to measure the weight of bacteria in the human body? I don't know. Then the second sentence they normally say is that the number of bacterial cells is 10 times higher than our own human cells, and the third one is that that microbiome actually encodes about 100 times higher number of genes than our own genome, which has 30,000 genes. Actually, that one I do know where it comes. Is from the first metagenomic study of the human gut, in which they only sampled 2 individuals. And now I have recalculated this with the data we have today, and including also data from the, all the bacteria living in the oral cavity, in the skin, and so on. And actually, it's about at least 1,000 times higher. So this is just to show you really that the metabolic, the potential of these microbes to encode different functions is enormous, and when you look yourself at the mirror, you should start to consider yourself more microbial than human, I think. So what do they look like? This is a picture, very recent picture from the last months, from a dental plaque, in the oral cavity of a healthy individual. It was made with a new technique called the classy fish, in which you label each individual species with a probe, which has a fluorescent marker. And in the past, we were able to do this with 2 bacteria, perhaps 3, but now with the new classy fish, you can do this with tens of bacteria. Here we have 10 of them. Each of them is marked with a different color. And it's just unbelievable. This is what happens in your mouth when you don't brush your teeth for 12 hours. And each color here is a different species. So the one here is Corynebacterium, and they appear to be the nucleus, the core of the dental plaque. From the tooth surface to the outside, facing the oral cavity. So all these bacteria here attach to those filaments at the edges. And those ones here, these guys appear to, they don't mind very much if they are in contact with oxygen. And then there are others here in the middle, more protective, the anaerobic species, and they seem to be highly structured. I mean, isn't it beautiful? Doesn't it look like a microbial Amazonian rainforest? It's really incredible. Actually, some bacteria appear to attach here, like Streptococcus, for example, and others appear to attach on top of those. So they are highly organized, and we are here seeing actually a highly organized microbial architecture. So there are all these interactions between different species of bacteria, but also between fungi and them, because we always talk about the bacteria, but we forget about fungi, viruses, and so on, and they actually interplay one with another, and in very complex interactions that we are starting now to understand. So, for example, here we have a hyphae of a yeast, here, and then on top of that, we have these different cells of Streptococcus that actually attach to specific receptors in the cell wall of the yeast. And I had the privilege of watching a film that was done a couple of years ago by Bastian Krom in Amsterdam, in which he had a film with the yeast, and then on top of that, there were the Streptococcus cells. And then we could also see the human immune cells, and they were moving around looking for some bacterial cells to eat or wherever they make for a living, you know. They were moving here and there in a random fashion. And then as soon as the immune cell actually touched the hyphae of the candida, as if it knew that the bacteria were going to be on top, it used the hyphae as a highway, and suddenly in less than a second, it just swallowed like tens of cells that were there. It actually, you know, it was something like this Pac-Man game I used to play when I was a child, and I was amazed. So the impression that gave to me is that the human immune cells actually were using the yeast as a cue to know where to look for the cells. And we consider these organisms as, you know, pathogens and so on. What I just want to know is that this illustrates very well how little we know and how the microbiome is the product of the interaction between all these players. And what is one of the main problems to study them? Well, that many of these bacteria, we have not been able to culture them. Here we have the tree of life for bacteria and archaea based on the 16S gene with all the phyla that were known a few years ago. And all the phyla which are marked here in white are those for which we have not been able to culture any single member of the whole phyla. And the poor guys, they don't even have a name, you know, they're called TM6, OP8, WS3, because you need to culture them before you can give them a name. So most of it we don't know, and what they do, you know. We just know that they are there because we have looked at their DNA. So the new era of the different omics studies actually allow us to know who is there, what are they doing, and so on. We are in a really exciting moment. So because we are studying mainly the DNA, RNA, and so on to know what are they doing, I had this very idealistic view of what my life was going to be. I thought it was going to be like the classical, you know, people working in the field 100 years ago, looking at the bugs through the microscope and having agar plates with the bacteria in a bench full of stuff. And actually my life most of the time is like this. I know it looks very boring, but it doesn't have the glamorous life I expected. But I'm going to try to convince today that actually we are in the middle of a really exciting moment, and if all these people, Pasteur, Cox, and so on, were here alive today, they would be absolutely amazed by the amount of information we can have. We are starting really to unravel what is their role in health and disease. These are some of the omics kind of methods that we use to study it. So if you just want to know who is there, it's enough if you amplify the 16S gene by PCR, and then we sequence it in a massive way, and we compare with the databases and we know who is there. But if we just sequence the total DNA in the community without amplification, we just have the total genetic repertoire of that sample. But then if we want to have a more functional approach, what we can do is that we extract the total RNA and we sequence it. And that way we will have access, we will know which bacteria are actively growing in the community, and also which are the genes which are being expressed at the particular moment. And the same if we do this for the total protein content or the metabolite content, we would be looking at the metatranscriptome, metaproteome, or metabolome of the sample. And okay, what can we learn from this kind of studies that have a clinical implication? The first thing is to detect, we can detect actually new pathogens. To illustrate this, let me show you this picture from a paper from a work we did a few years ago, in which we analyzed blood samples from people that had their wisdom tooth that have been removed. You know that many times when you do this, the dentist gives you antibiotics to prevent, you know, because some of the microbes in your mouth can enter the bloodstream, and that can be dangerous. So how do you know that you have some bacteria in your blood? Well, you culture them, and this is what we did for half of the sample. So we took half of the sample from each person, we cultured it under the different media, and these are the four genera that we found. The most common one was Streptococcus. The size of the word here is related to the proportion that we found of that bacteria in the sample. And then the other half of the sample, we extracted the DNA and we sequenced it, And this is what we found. We had an enormous number of, of microorganisms that we couldn't see before just by culture. We were totally biased in the microbes that were present there. Actually, some of them appear to be very common, and we need to know whether they can be a real threat. And then also look at this one, Streptococcus, that appeared to be the most common in the culture, actually occupies only in a small portion of the total. So we were totally biased because in the culture you allow the growth, of some specific microbes that are able to grow well under those conditions, but we were missing a big part of the picture. So we were only just seeing, well, the tip of a huge iceberg. And now, if we use this in any kind of samples, like from the eyes samples, from the gut, the oral cavity, and so on, if we look at specific samples and we can see new pathogens, I think we are really in the middle of a very big change that is really helping us to improve the way we do research and the way we diagnose and we treat many infectious diseases. So, for example, if we want to determine what are the causing agents, we have done this in some cases. In one of them that we have worked a lot is to study dental caries, which is considered to be the most common infectious disease in the world. It affects between 80 and 90% of the population, and is caused by bacteria in the oral cavity causing acid, and that acid breaks down the tooth tissue. So what we did was to take samples from the dental plaque of healthy teeth and also different caries lesions at an initial stage, and then in the dentin and deep dentin cavities. And we extracted the DNA, did a PCR of the 16S and massive sequencing, and this is what we get. What we got. What we have here in the x-axis is the sequencing effort. The more we sequence, the higher number of species that we see, until there is a point in which it doesn't matter how much we sequence, we don't see anymore. And what we find here is that in the dental plaque from the unaffected healthy teeth, we see that the number of bacterial species is about 600. And then when we take samples from the cavities, it's only about between 100 and 200. This is because that is a very acid place, and now some bacteria become dominant and are able to displace the others. But even if we look at the most extreme case here, the enamel caries lesions, the number of we have over 100 species there. So we are not seeing at a classical disease where we have an infectious disease with a single causing agent. Here we have clearly a polymicrobial disease, which makes it more difficult to study and also to treat. But then some people told us, perhaps part of the microbes that you see in your sample are not active. They just happen to be there. They could even be dead, because you could amplify by PCR DNA from dead cells. So we said, that's true, let's look at the RNA to only focus on the bacteria which are active. So we extracted the RNA, treated the sample twice with the DNase to remove all the remnants of DNA, and then we sequenced it. So we could look at which bacteria were active and the genes that they were expressing. So we looked at the metatranscriptome. And with this trick, we could tell apart who is there and only there and who was actually doing the job. I compare this, imagine that you are in the parliament, and we have the metagenome would be all the people in parliament, and the metatranscriptome would be only the politicians who are doing the work. So by doing this, we look at the DNA and the RNA, and then we are able to know which are active. So we did this, and actually, we saw that we have a very high number of species on each of the cavities. And the worst thing is that it varies a lot between individuals. Each individual has a different set who are causing the disease. And even within the same person, this is the most extreme case, where we have two cavities in the same tooth in a molar, and actually the composition changes between them. So here, the most common, is S. mitis, whereas here is S. sanguinis, and then we have this species here, which is absent there, and so on. So that means that probably for years, many people have been trying to do an anti-caries vaccine, and they don't work very well because they are focusing on a single individual, individual species. Normally, the Streptococcus mutans, which now we know occupies only about 1% of the total. So that's probably why all these passive and active immunization strategies are not working properly, and now we know why. Okay, another thing that we can check is, it's very important to know the microbes which are present on each of the different niches in the body and their health and disease, so we can actually know what is their function. When we look at, when we plot it like this, each point here is a particular sample, and we actually see that the samples from the same niche actually cluster in the same place, meaning that they have the same composition. We see here, for example, that the bacteria in skin are very different from the ones in the gut, or very different from the ones in breast milk, which also appears to have a high number of bacteria that we still don't know what is their function, but we believe have something to do with the modulation and the training of the immune system of the baby. Actually now, if you give me a sample from a human part and I don't know where it comes from, just by doing the analysis I can tell you exactly where it comes from. Although sometimes I have to say that we have some unexpected surprises, and of some cells that are in the place where they shouldn't be, Anyway, I leave it there, but sometimes it is amazing what people have in their mouth. The other thing that we have to keep in mind is that we are losing a lot of microbes, especially in the developed countries where, everything is extremely clean, including the food that we give our babies, and mainly also because of the overuse of antibiotics, we are losing a lot of our microbes, and that can have very important consequences. In fact, many scientists believe that the increase in all these kind of diseases which are related to the immune system, like autoimmune disease, allergy, asthma, and so on, could be partly related to the decrease of the microbes in which our children are exposed. And if you are interested in this, since we are in a library, I highly recommend reading this book, which talks about this topic. Also, they can provide some protection against pathogens. We all know that when we have antibiotics, you can have a secondary infection by opportunistic pathogens and so on that now find the niche free. We can see this in samples that we took a few years ago they were biopsies from the stomach in individuals with and without Helicobacter, which is, as you know, the causing agent of gastric ulcer. And when this species is present, what we see is that actually most of it is occupied by it. Between 70, 80, in some cases 90% of the bacteria in the stomach are actually Helicobacter. Whereas when you don't have that in a healthy individual, we have a more diverse ecosystem. And some of these actually we have found some lactobacilli that appear to have an antagonistic effect against Helicobacter. So this is one of the things, everything is under balance, and some of the microbes that we have are able to prevent the infection by other pathogens. Of course, we need to understand the interaction with the immune system. This has a lot of clinical relevance. This all began by a sort of a dream, that we had in the lab a few years ago that appeared to be like science fiction, if we could tell apart, which of the microbes are actually able to be recognized by the immune system, like these ones, and which ones are able to evade the immune response, to evade the antibodies. Well, now we have been able to do this by the use of flow cytometry, and what we do is to which is actually a microfluidics kind of apparatus in which the cells are passed one by one in a line, really in a row, and then you are able to detect them with a laser. And what we do is that we mark the DNA of the cells with one marker in red, for example, so we really know that we are looking at real cells. And then with another marker in green, for example, we mark the antibodies, IgA or IgG or IgM. So if we have cells which are marked in red and green, we know that those are cells which are are going to be coated by antibodies. And if they are only marked in red, then we know that they are only bacteria and they are uncoated, so we know that they are not recognized by the immune system. We have done this in different kind of samples, in saliva, fecal samples, and so on. Then actually, if you also have a sorter, and this is quite amazing, you're able to separate the bacterial cells one by one according to those colors. So we can put in a tube one bacteria that is going to be coated by IgA, for example, and then if the next cell is only marked in red, we know that it is not coated with Igs, so we put it into another tube. And this really is like an act of faith, because this happens at the rate of several hundreds per second, and it's just amazing how it works. Then when you look at the tubes, you see that one is red and the other one is green, so it really works. What we do, when we have the two tubes, we amplify by PCR and sequence. So we know which are the cells which are seen by the immune system and which ones are not. We have done this in different cases. This is our last work, in which we looked at 1-month-old fecal samples from babies that belong to a Swedish cohort that had been followed for 9 years. So they were samples collected 9 years ago. And actually, they followed those children, and they know which ones later on in life, when they were 5, 6, 7, developed asthma, allergies, and so on. In this case, it was asthma. What we have here, this IgA index, it means that if the values are positive, it means that this genus is actually always found coated with IgA. If the values are negative, it means that the bacteria are free, they are uncoated by antibodies. And as you can see, for example, in healthy individuals, E. coli is always coated by antibodies in these 1-month gut samples, whereas in children that years later are going to develop asthma, they already have some problem because they are not able to recognize E. coli, and the opposite pattern can be found for some other species. So this is quite interesting because we can predict in a 1-month-year-old baby that is going to have a very high risk to have asthma in the future, perhaps when that baby is 4, 5, 6 years old. And this is a really powerful measure, to diagnose the risk, so we can, take some treatments in between to delay or to prevent that this is going to happen. And this takes me to these 6 clinical implications, the diagnostics. We have also done the same with people suffering from colorectal cancer. And what we can see here is that biopsies from the control individuals without the disease are very different from the bacteria that we find both in the tumor and in the polyp before it is going to develop into the tumor. So just by looking at those microbes, they can serve as biomarkers of the disease even before the first clinical symptoms appear. So this is very promising, and I believe in the future this might be very common in hospitals. I can see a future where we can have a blood test or a urine test, and also, why not, a gut or oral microbiome test. And if we contrast that with the data that we have, we would be able to predict whether we have a high risk of having a disease, or whether it is already indicated that we have that disease, even if there are no clinical symptoms. And finally, and this is the most applied research that we have been doing, the more advanced of the one that we have, is the search for new bugs that we can incorporate into people and that are supposed to improve their health.
2 minutes.
What we call, okay, yeah, I'm at the end. So what we did here was to extract the total DNA from carriers-free individuals, an individual that that were carriers active, and then we extracted the DNA and looked at the functions and who was there. And we were quite struck by the fact that carriers-free individuals had a very high frequency of what appeared to be a new species of Streptococcus. And so we thought that perhaps it could be having some function to prevent the appearance of dental caries. And so we went back to those individuals and cultured them until we actually got it. All this came from one of my lab mates in the lab 6 years ago, that told me, I am one of that 10% of the population that never had dental caries in their life. I have never had a single cavity, and my boyfriend used to have, but after he is with me, he's no longer having more dental caries. So I think I'm passing something to him. And I was like, oh, wow, perhaps, in the kissing, she's actually passing to him some of the microbes in the saliva, and that's going to have a protecting effect. So she was our first patient, and it is true, she had a very high frequency of this of the microbe, and our surprise was when we culture it in the presence of the bacteria causing dental caries, it actually kills them. So here we have a long culture of some of these ones that cause dental caries, and in the middle we have our anti-caries bag, and we see that there is an inhibition ring around it. So this was a really exciting moment in my life when we saw this, that the people without caries actually have some microbes that are able to protect them from the disease. We sequence it and so on, and happen to be a new species that we have called Streptococcus dentisani, from healthy teeth. It looks as beautiful as this. And this is how it kills S mutants and the other ones. It makes these pores here because it produces antimicrobial peptides that make a pore in the cells, and then it kills them because they cannot keep the balance between the inside and the outside. All this water coming in, and then they lie like this. And here we have the dentisani killing S mutants. We are now starting the clinical trials after a very long process. So this has been quite a lot of work, but we are very exciting. We also just finished a scale-up because our idea is to have this very cheap so it can be available for most of the population around the world. And now we actually have managed to produce it at the price of several cents of $1 per dose. So we are very happy with that. We are going to have two these product lines. One that the dentists who are not very happy with me actually can put in the clinic, and then other that people could just buy in the pharmacy that we still don't know what's going to be, but probably some chewing tablets or something like this. So in conclusion, the different methods that I have shown you, I think they all, provide some kind of information, and all of them are important to really understand, what is the role of these microbes in health and disease. I think they can be used as a marker of the disease, and I think this could be done in hospitals in the future. I think we are also killing our microbes and the functions that they encode, and this can be important. And something that we knew already from the point of view of love, but now we can take that, science also says the same, that kissing can improve your health. So thank you very much for your attention, and I will take any questions.
Well, thank you very much, Alex. This is very, very interesting. Unfortunately, we have to move on as quickly as possible. They told me they're going to kick us out if we don't finish on time. So Dr. Mir is going to be there, and please talk to him after we're done. Because this is very, very exciting. So we'll move ahead to the next speaker, Dr. Bent Peterson, who comes to us from Denmark. He is an associate professor in the Center for Biological Sequence Analysis in the Technical University of Denmark. He has years and years of experience with bioinformatics, computational biology, sequencing, metagenomics, as well as genome assembly. He worked on many, many exciting projects, and he was part of teams that sequenced genomes from horses, ancient horses, extinct animals, malaria, as well as a lot of metagenomic samples. He published in many high-impact journals such as Science, Nature, Cell, PNAS, and in 2014, he started his own company, which offers a lot of extensive and computational-heavy bioinformatic analysis. So, Dr. Peterson, the floor is yours.
Thank you very much for the nice introduction, and thank you so much for inviting me here. It's an honor to be here in the Library of Alexandria. I don't know if you noticed before in Dr. Rami's talk, he was referring a lot to this will Ben talk about, this will Ben talk about. So actually, it was difficult for me to find a title for this talk because one, I had to talk about microbiomes, I had to talk about our computational infrastructure, I had to talk about our genomes, and now I also had to talk about assembly. But unfortunately, assembly is not
in 25
here.
minutes.
But I will do my best. So I'm a bioinformatician, and today it's very good to be a bioinformatician because we have so much data from so many fields. What I'm working with is in the genomics and the transcriptomics section. Years ago, bioinformatics was a relatively small area. It was very difficult to get enough data because it was very difficult. As you can see, the price for a human-sized genome, of sequencing of a human-sized genome in 2001 was close to $100 million. And then something happened in 2008, and this is when we got the first next-generation sequencing technology. The price dropped significantly, and suddenly we were able to get a lot of data, and it's getting cheaper and cheaper. This you can also see here. We have a so-called sequence data avalanche. The blue line is the computer annotated sequences, and you can see it's clearly growing much faster than the manually curated SwissProt and PDB, because we simply don't have time to look at it in a lab. I think it's very interesting that sequencing today is rapidly evolving in both the sequencing technologies. It's getting faster, you get more accurate data, and it's getting cheaper. There are a whole community developing better and better tools, and now it's getting a routine, as you heard before, for biological research and applied research. I also think it's very interesting that it's becoming a part of personal life. Non-scientists are getting a DNA test to see if they have health risk or to find distant relatives in other countries. And you have companies starting to offer genetic tests, for example, for fitness and etc. So one of the projects I will talk about is a project our group was involved in, where we had both microbiomes and a lot of data. And there's a plane here as a hint. Now I will show a small animation. Is there any of you that know this game? I don't see any hands. It's a game called Plague Int from Endemic Creation, and it's actually a quite cool game. Your task is you start as a pathogen of your choice. You could be a fungi, you could be a bacteria, a deadly virus, and then it infects patient 0, and then you have to keep evolving the pathogen with the end goal of wiping out the human population. It's actually fun. It's challenging, but it's also very scarily realistic. You all know about the Zika virus. Infectious diseases are the cause of about 22% of all human death, and today, as you know, with Zika, international flights are a major cause of transmission. And today the problem is that international disease surveillance is based on doctors' reports. And one problem with that is that it's only when you feel sick and you actually go to the doctor that the doctor starts looking at it and may make some correlations. Another problem is that the clinical microbiology today is not geared for rapid detection of an emerging pathogen. Depending on the complexity of the sample, it can take weeks to months to just get the samples to grow, and it can take even longer to type it. But this is where sequencing steps in and can help us. So what they did in the project was that 18 long-distance flights from 9 different locations, all coming to Copenhagen. We had a deal with Scandinavian syst flights, where we got access to actually taking the samples from the planes. Planes were selected from North Asia, China and Japan, South Asia, Thailand, Singapore, Pakistan, and North America, New York, Washington, D.C., Toronto, and Greenland. And it was recently published in Nature Scientific Reports. So the project was that samples were collected from the airplane toilets. Each plane has around 400 liters of human waste. The idea is here that if you're on a plane and you're healthy, you may not necessarily go to the toilet, but if you're sick, there's a higher likelihood that a person will go to the toilet. So samples were collected from the storage tank, I don't know if it was a poor student doing that. I'm happy I didn't do it. The samples were brought to the research facility and it was sequenced. Then the aim was to look for pathogens and for resistance genes. At the first glance, the samples were quite boring to look at. We didn't find, it was lucky for the passengers, maybe a bit unlucky for the project, but we didn't find any nasty pathogens or nasty diseases. You can see that the samples look very similar in composition of bacteria, plasmids, and invertebrates, etc. When you look deeper into it, it starts getting interesting that we can actually cluster the samples based on the read abundance from each flight, so you can see that they're grouping together. If we look at a few selected pathogens, you could see that Salmonella enterica, which causes diarrhea, was more common in South Asia, and you can see that norovirus were more common in North Asia. Sorry, I mixed it up. South Asia before and North Asia. And if you look at the antibiotic resistance genes, you can see that in the South Asia, there is a higher abundance of antibiotic resistance genes, and it's less in North America. But this most likely corresponds to that it's easier to get access to antibiotics in South Asia, and in US, etc. It may take a visit to the doctor where they are not interested in prescribing it. This was done on 18 planes. If we were to do it on a global scale, it would require, first of all, a lot of money for the sequencing, but we would also need to have a lot of data, around 1 petabyte of sequencing data per day. In 2012, 1.1 billion passengers were traveling on international flights, and as you can see, it's only getting higher and higher number. So this is clearly, if you want to have it on a global scale, we need to have some sort of global funding from the countries, because I don't think a single project would be able to pay for that. So taking to the next step from the project, we are now going to look at global surveillance of diseases again and antimicrobial resistance genes from sewages. So right now, people are sending samples from more than 70 countries, more than 180 cities, to the sequencing facility at DTU. And then the goal is to understand the occurrence and burden of resistance in a defined human healthy population. Here it's opposite from the airplanes, because in the airplanes, we suspect that there are more sick people, samples from sick people. But if you look in a city, you should hopefully have more healthy people. But how do we handle all the data that we get from all these projects? Well, one thing is to write better algorithms. This is not something we focus on in the group, because there's a whole community doing that, and they're probably much better than we are. Another thing we can do is to start working with artificial intelligence. We are starting up on that now, and we have a history for doing machine learning. And then the next thing, which was our solution to the problem, is to simply just build bigger computers. At CBS, we have more than 20 years of experience with bioinformatics, working with supercomputers. One problem there was that it was funded by several funding rounds, so you bought some hardware, you bought some disk, and you bought some more CPUs. But it was not really optimized for life science. One problem when you have a lot of data, you want to assemble big genomes, is, for example, the I/O. You need to be able to read the data really fast, and you need to access the memory really fast. So this is what we were looking at. It took a year to design our new supercomputer, which opened officially in December 2014. It took a year to design and 3 months to build. So we have around 16,000 cores, all on a very, very fast bandwidth. It says that we have 3 petabytes of storage. We now have 4, I think. And then it's also a green facility, so I can, with a good conscience, start all 16,000 CPUs because the heat generated from the machine is used to heat the surrounding buildings. And I have a small video I would like to show you from the opening of the computer.
Every 6 months, the volume of digital data on living organisms doubles. Genes and proteins are described for single organisms and for entire populations in everything, from bacteria to humans. Vast amounts of clinical data from the healthcare sector are recorded and available in a digital form. With the help of
Powerful supercomputers, we can transform these data into valuable knowledge on complex diseases, more precise and cheaper medicine, or new biotechnological applications. Denmark has taken an important step towards making this happen with the investment in a life science supercomputer facility at DTU. The system, called Computerome, will enable researchers across Danish universities, companies, and healthcare institutions to collaborate on data and computational resources. It's also an important part of the European life science infrastructure, ELIXA, that connects Denmark with many of Europe's leading bioinformatics resources.
In order to play a role in today's biology and medicine with all those data, we really need capacity and computational muscle. And this new supercomputer is really giving us a unique position where we can attract data that we then will not have to pay for ourselves, and we can use them in our analysis and combine them with our own data.
The new life science supercomputer installation at Risø is very unique in the way that it is optimized only for life science applications. It means that bioinformatics research, healthcare and the pharmaceutical industry will have an opportunity to work in a collaborative manner on a number of the same curated datasets.
This new area, bioinformatics, systems biology, medical informatics, it's not just research, it's also business, because methods and solutions are needed. So there will be a lot of business development, a lot of new companies created in this domain that really spans different worlds, hardcore supercomputing with clinical and molecular biology domains. It's actually not so difficult to predict the future in this area because the data will become cheaper and cheaper, the supercomputers will become more and more effective, and we really need to put these things together in order to make treatment more precise. And I'm very optimistic because we have the means and we also have the needs. We need to make treatment more efficient and cheaper, but we will also be in a very good position to achieve that goal.
This was our little toy. One thing that Peter said in the video was that it was completely optimized for life science. And it actually makes a big difference because when I was assembling genomes on our old system, human genomes, it took around 3 to 4 weeks for one assembly, but on Computerome it only takes 10 days. So optimizing towards the data that we're actually using helps. So to quickly conclude, it is possible to track global disease outbreak using long-distance flights. And it's possible to use real-time monitoring from sample to result. It was around 2 days. Doing it globally would require that we have many petabytes of storage, and we definitely need bigger computers for that, and we would also need a lot of money. Then I also promised to talk about some of the funny genome projects that we are working with, the unique environments. And one of them here is the vulture microbiome. You all know that vultures, they eat decaying or dead animals. They actually stick the head into the animals, but they don't really get sick. So they have a very special microbiome in the face, but also in the gut. They have a very low pH in the gut, less than 1.5. So the bacteria that lives there has to be very special. Then we have the oriental hornet. I really like this one. It's a hornet living in Jordan, and basically it has solar panels on the back. You see the yellow stripes that any wasp has. In these stripes, they're actually able to convert solar energy into energy. We are currently looking into to figure out if it's the cells themselves or if it's the microbiome living on the wasp that is able to convert it. Then we have this sweet little guy, the koala. Koala are very funny animals, and they are very picky animals. They only eat eucalyptus. Eucalyptus itself is is full of toxins, and it's not very energetic. There's not a lot of energy in it. So the microbiome that the koala has is also very special because it's able to cope with the high toxic pressure, but it's also able to take out all the energy from the very low energy diet. I think then we have Arctic microbiomes. Our boss in the group, Thomas Sigurdspontin, went on a Galatea expedition with a research ship. They took samples from the polar sea from different depths. And we found many proteases that were still active at 5 degrees, a very low temperature. And this is something that can be used to improve washing powder, for example. And then I have a slide here just summing up some of all the other interesting projects. Here you can see this is a giant squid. The giant squid is very special. It lives deep underwater, several hundred meters depth. So the genome itself has to be, or the organism has to be special because it can cope with the high pressure. Then it's also suspected that when you see these whales, I forgot the name, and they have scrapes on their side, it's actually giant squids that are attacking them. I have some lips here, the red color. I don't know if you know it, but the red color that's used for lipstick, nail polish, or alcohol, it actually comes from a small scale bug that are grown on cactus. They put one male with 500 females. Then they mate and they reproduce. When you dry the organism, 25% of the dry matter is the precursor for carmine. Up here we have vampire bat. It's a project that first has to do with the actual genome of the vampire bat, but also the microbiome. As you know, bats are, you have bats that are feeding on fruits, and then you have the vampire bat. Vampire bat feeds on blood. Blood is, there's not a lot of energy in blood, and the bat is so special, so when it starts feeding on a cow or whatever it's feeding on, it actually starts peeing on the same time because it's excreting all the stuff that it doesn't need, namely the water, and then it's uptaking what it needs to survive from the blood. So here again, the microbiome has to be very special. And then one of the last projects here is wine. Before I started on the food genomics project, which has to do with wine and silage, I never thought about that wine is actually full of bacteria, fungi, and many other things. the company we are working with, Christian Hansen, they are selling starter cultures for vineyards to either give a certain flavor, but we have shown now in the project that when you inoculate with a specific lactobacillus strain that they lucky enough produce, then it's inhibiting, for example, Acetobacter, which is the bad guys in wine, and then it's increasing the growth of other lactobacillus. And then up here is a 700,000 year old horse, not this one, but a few years ago we were working on the genome of this very ancient horse. And that was all I had to say. Thank you for your attention.
Well, thank you very much, Bent. We have time for one or two very quick questions for Alex and or Bent. So if you have any question for the last two speakers, please come forward.
Yes. I want to ask you, I'm a dental student, so the moment of the sampling to determine the microbiome of the cavities, I was wondering about the pH and the temperature. It will clearly affect the type of, or the shape of microbiome at the moment of the sampling and from patient to another. So if you want to create antibiotics for those microbiomes, it will be so vary. I mean, there is so much variables in the oral cavity because it's unstable environment. So how can we determine that this microbiome, what's the cause for that cavity?
Sorry, I don't know if it is because in here I cannot hear you very well. So your question is whether how can I be sure that the microbes that we see are the ones causing the disease?
Yeah,
because there are many factors involved in the sampling, like the pH, temperature, and so on. Yeah, I mean, it can have an influence on the microbes that you have, but I guess what we do, for example, if we take dental plaque samples, it varies a lot, so we always take samples with the same number of hours since the last toothbrush. They are all, for example, 12 hours oral samples or 24 hours. In the case of the cavities, we don't do that. And I guess, the pH, you mean it is going to vary during the day, or, for example, that after lunch you can have a change and things like that? Yeah, it is true. That will have an effect if you look at the RNA. So, for example, we have to look at the bacteria which are active before and after a meal, and they do change. But when we look at the DNA, it doesn't have an effect because the DNA just have everything that is there, and some of them are going to be more active at one particular point and others at some other point, but the DNA appears to be more or less stable. So I think there, it may change, but I think it would be impossible to select always the same kind of samples. It's impossible to control all these factors, We have seen, for example, when we took samples of saliva, it does vary through the day. So the saliva at the beginning of the day, for example, is more dense, and then it becomes more liquid and so on, and it actually affects what you see. So in that particular case, it is important to take always the same kind of sample. For example, you can collect samples just by drooling or by spitting or by using chewing gum to produce more saliva, and all this actually change what you see in there. So with the saliva, we know that we always take the samples at the same time, and with the dental plaque samples, we normally take always in the morning and with a given time. With the cavities themselves, we have not looked into how it actually changes at different times of the day and so on. So they might vary, and you are right that it might be a factor introducing some noise. I agree.
Okay. I have a question for Ben. For your project working with sequencing the microbiome of international travelers, Could you elaborate on the advantage of using a sequencing approach as opposed to a hybridization-based approach, like a fellowship or ship-based approach, which might not need all of the storage data, storage capacity?
How does it?
You get it.
One advantage of sequencing everything, a whole genome, is that you can also, if you want to look deeper into some samples that you get, then you can do the assembly and look into that.
Okay,
Yeah.
thank you.
I also have a. So does the airplane food affect? Like that? Does it depend on the food or the country they came from? Did you know the answer?
That I don't know.
Oh, okay.
We could see that from Asia they were eating more nuts.
Okay.
But if it's from the food, I don't know.
One more question.
Well, I guess it would not be from the food you get on the airplane, because that would not come out in the same flight. So.
You never know.
I have a question.
Dr.
please.
Yeah.
So, I'm sure you thought about this, that something that the Zika a mosquito can feed on in a way that it actually destroys itself. Is this something that somebody's working on to get rid of that Zika virus pandemic?
The Zika mosquito?
Well, there's no Zika mosquito.
No,
is
not
there?
a mosquito. I mean, it's something that the mosquito will be feeding on a Zika
You have an answer?
thing that actually kills
I don't know.
the mosquito or kills the...
Maybe Guilherme knows. I don't know. Do you have an answer?
Does somebody?
Okay, the only thing I can add to that is that on the Wolbachia infected mosquitoes, they don't take up the virus as effectively as the ones without Wolbachia, so it's one way of blocking it. And that's been, in Brazil now they're releasing these mosquitoes as well as transgenic mosquitoes. Well, they can bite people with Zika, but the Zika they don't pick up it effectively.
Thank you.
No. Can I ask a question?
Yeah, sure.
So that's for Bent. So I agree with what you mentioned about increasing computer power and going to AI, but I would also argue that getting programmers to do some hardcore optimization will be useful for two reasons. One is that Moore's law is gone now. We cannot keep on increasing compute power at the same speed and cost that we could. And the second is that there's one good example by optimizing GATK. Intel and the Broad guys were able to increase 1,000 times the speed at which it processes the same program. So I'd just like to make that argument and have your thoughts on the future of hardcore compute power.
Well, I totally agree in that, and one thing that we are lacking in the community, or at least at our group or the university, is that we have some people that knows how to code, because we are doing codes and we are doing pipelines, but we are maybe not doing it optimally, or we are definitely not. So having a scientific programmer is definitely something that could make things run faster. Yeah. And this is something we are also starting in the group to having scientific programmers writing our code for exactly that reason. I agree.
A question to Dr. Rami. Do you have any experience regarding using the high performance computing facility at Bibliotheca Alexandrina through the Link Scheme project? Link Scheme project is a common shared project about the countries of the Mediterranean, and there is an open access facility here at the Library of Alexandria. I don't know if you performed any bioinformatic analysis through this facility or not.
I'm actually, I'm not familiar with it, so I would like to know more. Maybe, yeah, of course, the library, they help us in every aspect, so that would be good too.
Well, thank you very much for attending, and I guess we'll see you all tomorrow.